Good morning everyone. Today I am explain about introduction on nucleic acid. Nucleic acid is a macromolecule with acidic property and it was isolated from the nucleus of a cell and hence it is called as nucleic acid. It is made up of carbon, hydrogen, oxygen, phosphorus and nitrogen. This nucleic acid was first isolated by mixture in 1868 from the nuclei of pus cells on hospital bandages. Mixture named it as nuclein, then later Altman in 1889 named it as nucleic acid. Occurrence of nucleic acid. Nucleic acids are found in all organisms such as plants, animals, bacteria and viruses. They are generally found in the nucleus as well as in the cytoplasm of a cell. General structure of a nucleic acid. Nucleic acid is a long chain polymer and composed of monomeric units called nucleotides. Nucleic acid is a long chain polymer composed of monomeric units called nucleotides. Each nucleotides consists of a nucleosides and a phosphate group. Each nucleotides consists of a nucleosides and a phosphate group. Next, each nucleosides consists of a pentose sugar. Each nucleosides consist of a pentose sugar, that is 5 carbon sugar and a nitrogen base. The sugar is ribose in case of RNA and deoxyribose in case of DNA. The nitrogen bases are of two types, namely purine and pyrimidine. The purine contains two main bases, that is adenine and guanine. Similarly, pyrimidine contains a three main bases, that is cytosine, thymine and uracil. Cytosine and thymine are commonly found in DNA and uracil is found in case of RNA instead of thymine. Next one, nucleosides. Nucleosides, a nitrogen base combined with a sugar molecule to form nucleosides. In deoxyribonucleic acid, 4 different types of nucleosides are present, which are as below: adenine, guanine, cytidine, thymidine. In case of ribonucleic acid, deoxyribose sugar is replaced by ribose sugar and thymine base is replaced by uracil. In case of ribonucleic acid, deoxyribose sugar is replaced by ribose sugar and thymine base is replaced by uracil. Nucleotides. Nucleotides are defined as phosphoric acid ester of nucleosides. A nucleosides molecules combine with a molecule of phosphoric acid to form a nucleotide molecule. The deoxyribonucleic acid contains 4 different types of nucleotides, which are as below: adenylic acid or adenosine monophosphate, guanylic acid or guanosine monophosphate, cytidylic acid or cytidine monophosphate, thymidilic acid or thymidine monophosphate. The ribonucleic acid contains uridylic acid or uridine monophosphate instead of thymidilic acid. Next one, composition of nucleic acid. Nucleic acid consists nucleosides and phosphoric acid, and nucleosides consist of nitrogen bases and sugar molecule. Nitrogen bases are two types, purines and pyrimidines. Purines are adenine and guanine. Pyrimidines are cytosine, thymine, and uracil. Sugar molecule are of two types, ribose sugar or deoxyribose sugar. Nucleic acids are broadly classified into two types based on the types of the sugar present in them. First one, deoxyribose nucleic acid and ribonucleic acid. Chemical composition of deoxyribonucleic acid, that is DNA. DNA is made up of three important chemical components, namely sugar, deoxyribose sugar, and phosphoric acid and nitrogen bases. Sugar molecule. The sugar present in the DNA is called as deoxyribose. It is a pentose sugar which contains the 5 carbon atom C5H10 and O4, and it contains one oxygen atom less than the ribose sugar at the carbon number 2 of deoxyribose, that is only HCH, that is CH2 group is present, but in case of ribose sugar second carbon atom contain HCOH group. See the structure of deoxyribose sugar. Next, phosphoric acid. Phosphoric acid links the nearby nucleotides by joining their pentose sugar with a phosphate diester bond. This bond links carbon 5 in one nucleosides with carbon 3 in next nucleosides. In the diagram, phosphate group and deoxyribose sugar linked with the phosphate group at 5th and 3rd position. And nitrogen bases. Nitrogen bases are nothing but nitrogen containing organic compounds and are of two types, namely purines and pyrimidine. Nitrogen base molecule is joined to sugar by glycosidic bond. Purines are two-ringed N2 compounds. They are of two types, namely adenine and guanine, and pyrimidine are single-ringed N2 compound. They are of two types, namely thymine and cytosine. In the diagram, adenine, guanine, cytosine and thymine structures. Thank you.